Hello, thank you for joining us. My name is Sven Bocklandt, and I head up scientific affairs here at Bionano Genomics. I'm really excited to talk to you today about our optical genome mapping technology. And as the title says, we're calling it the beginner's guide because we're really going to touch a little bit on many applications without going in too much depth on any of them. If you've already watched our symposium, all of our symposium talks about a month ago, then I'm not going to show you much new. But if you're new to the platform, then this should be a great introduction. I would like to start with this quote from our Nobel Prize winner, Sidney Brenner, who said that progress in science depends on new techniques, new discoveries, and new ideas in that order. I'm going to talk about a new technique and show you many new discoveries made with this new technique, and I hope that you come up with new ideas of exciting studies or research you can do using our platform. Now, I want to start by pointing out that optical genome mapping with our Sapphire instrument is not a sequencing technology. We cannot detect a single nucleotide variation in the genome. We simply don't see that. What we do detect, and we're really good at, is structural variations, starting at 500 base pairs and then up to all the way entire chromosomes, aneuploidies. And within the structural variations, our platform can really detect every single type of structural variant. And I will show you many, many exciting examples in the course of this presentation. Now, if you're thinking structural variation detection, maybe you're thinking of exciting new methods that you have heard talks about or have seen at conferences, but really the standard of care in the clinic to detect structural variations in patients with cancer or genetic disease is this: it's karyotyping. And what you do in karyotyping is you use a microscope to look at intact DNA molecules. So you break open the cells and look at the chromosomes with a microscope. And you visualize patterns on these intact DNA molecules. And when you see changes in these patterns, for example, a pattern is flipped around, or a pattern moves from one chromosome to another chromosome, then you detect that as a structural variant. That process is not manual, takes a lot of time, but that is still the standard of care. Now, why is that? Why hasn't sequencing replaced all of that? Well, this is one of the reasons why. If you run an NGS experiment, if you're using short-read sequencing to look at the genome, the first step is fragmenting the DNA. Here's a graphic from a company that produces these tools that fragment the DNA that proudly show that nothing here is left over 300 base pairs. The entire genome, with its perfect, intact, beautiful structure, gets fragmented into tiny little bits that you then put on your sequencer and analyze. And then after you've destroyed that structure of the DNA, you try to piece all these little fragments of a couple hundred base pairs together, trying to figure out what the structure was before you destroyed it in the first place. It just doesn't make any sense. And the other problem with NGS is that the human genome is just highly repetitive, with just about a third of the genome consisting of unique sequences and 2/3 consisting of repeats. So if you can only see a couple hundred base pairs far, as you do with NGS, then you can't make sense of this 2/3 of the genome that is repetitive, because if you get sequence back from, say, a line element, which of the 100,000 copies of that line element that you just sequence, it's impossible to tell. And maybe you think, well, I don't care about repetitive sequences. I only care about the genes. But what happens is that a lot of the structural variants in the genome are flanked by repeats and are actually caused by the recombining of these repeats in the first place. So if you're blind to this 2/3 of the genome, you're also blind to the structural variants that are flanked by them. And that's why karyotyping is still standard of care for clinical detection of structural variants. Now, Bionano uses the same principles that you see here. We also look at intact DNA molecules, and we also visualize patterns on these molecules. But what we're adding are our nanochannel arrays. You see an electron micrograph here of one of our chips, and each of these little channels that you see fits a single DNA molecule. And instead of the 500 to 800 bands that you can see in a karyotype, we place 500,000 bands, if you may, on these DNA molecules in the form of fluorescent labels. So massively parallel imaging of ultra high resolution DNA molecules. That is really what we do. And our microscope is called Sapphire, and it automates the electrophoresis and imaging of these DNA molecules. Now, we don't just make the instrument, we also make all the components for the entire workflow. So that includes the chips that contain the nanochannels. We make kits for DNA isolation and labeling. And then we have our own software called Bionano Access that you can use to detect these structural variants. Now, let me tell you how it works, and I'll try to do this as quickly as possible. The first step in imaging megabase-sized DNA molecules is to actually extract these ultra-high molecular weight DNA molecules. And that is not obvious because every standard DNA isolation protocol, whether you use spin columns or magnetic beads, or you simply precipitate the DNA, they all break the DNA in fragments of maybe maximum 30 kb. And that's just not enough for what we want to do. So instead, we developed our own protocol where you gently lyse the cells in solution and then grab onto the DNA with a silica disc, similar to the material in a QIAGEN column. And then you wash and elute the DNA in a clean tube. So now you have ultra long DNA molecules that have not been exposed to any mechanical forces, so they're intact. This protocol takes about 4 hours, is automatable, and we can work with fresh or frozen blood, with bone marrow aspirates, with cultured cells, or with fresh or frozen tissue. Keep in mind that we cannot work with cancer samples that have been fixed in FFPE because the formalin fixation process really destroys the long DNA molecules. Now, the molecules that we isolate are extremely long, as I mentioned. Here you see a good but not exceptional run of a bone marrow sample on Sapphire. And you can see that the weighted average of the molecule length is about 400 kb here. We see at least 300 kb molecules. And then the tails off all the way into megabases. Here's 1 megabase, and so this entire section are molecules longer than 1 megabase. Compare that with long-read sequencing, where with PacBio you get about 12 kb reads, and with nanopore sequencing, you still get in that same range, orders of magnitude shorter than with Bionano. So now with megabase-sized DNA, the second step is to add a label. We're staining the backbone with an intercalating dye that provides the blue signal so we can see where the molecules are. And then we add a fluorophore at the position of a 6 base pair sequence motif. We use the same 6 base pair motif all throughout the genome, and it occurs about every 5 kb or so in a human or mouse genome. And now at the end of that process, which is a very simple single enzymatic reaction, we end up with something that looks like this, megabase-sized molecules with green labels attached. Now, this is the core technology that our company was founded on. The ability to get these DNA molecules to squeeze into nanochannels. Each nanochannel fits a single molecule, and when the molecule is in that channel, it is uniformly stretched. And because of that uniform stretch, we can get all the information that we want just by taking a picture. Here on the left, you see a beautiful video of the DNA moving through pillar features from the bottom to the top using electrophoresis into these channels. Every vertical line that you see is a single molecule of double-stranded DNA. And from here on out, every line that you see is a molecule that sits in one of these nanochannels. So this is a process of moving the molecules into the chip and filling up these nanochannels. Again, here the DNA is coiled up and then moves into the channels and linearizes. On the right, again, you see the actual chip, and you can see how each of these DNA molecules sits in one of these channels. Now, the instrument will cycle molecules in, image them, pull new molecules in, image them, and do this over and over again. And that allows us to collect as much as 100x coverage on 12 human genomes per day per instrument. That is an enormous amount of data in a short amount of time. Now, if you need more data, say to analyze a cancer genome, then you can simply run that chip longer. It doesn't cost you any more in reagents if you have the instrument, You simply run it longer to collect as much as 400x on 3 samples per day per instrument, or you can even continue and run 1600x coverage on 3 genomes. This costs just about $500 or so per sample. We can talk about that later, but at 1600x coverage for $500, that is just completely unheard of, an enormous amount of data. Now, once the instrument images these DNA molecules, we have a couple different workflows that we use for different applications. So what we can do here is, if you're looking at a genetic disease patient or say a cell line, a monoclonal cell line that you're studying, what we will do is we will extract the label patterns on each of these molecules and perform a complete de novo assembly of the genome. De novo assembly means that we pairwise align every molecule that we imaged with every molecule that we imaged and build these overlap graphs that you see here. And then out of the overlap graphs, we will calculate the average spacing between each of the labels and build this consensus genome map that represents the average spacing. And then we call structural variants by comparing these patterns with the reference. So if the green block here is the reference genome, we know what the sequence is, and therefore we know the position of that sequence motif that we labeled. And then we simply align the map of your patient sample to that reference genome, and we look for differences. In this case here, you see a deletion of the reference. Now, using that same mechanism, we can detect all classes of structural variants. A deletion here is called when the distance between labels, this label and that label, decreases by more than 500 base pair. We call that automatically genome-wide as a deletion. When the spacing between 2 labels increases, we detect that as an insertion starting at 500 base pairs. Now, if there's more of a pattern that we recognize, then we can see that a certain pattern present in the reference aligns twice in the same map. We'll detect it as a duplication that can be direct or in tandem starting at 30 kb. An inversion is picked up when a pattern is flipped around, and then a translocation is detected when a de novo map in your sample aligns for half with one chromosome and half with another chromosome, or a different part of the same chromosome. And then there's repeats that we can detect as well. In some cases, there is a single label for each repeat unit, but when that's not the case, we can simply measure the flanking label. So for example, if we know that there's a repeat array here, we can measure the distance between this and that label, and you can see that that expands significantly. I'll show you some examples of that. Now, we also have a copy number variant tool, and that works differently. That simply aligns the molecules to the reference and counts them and normalizes them. And when there's an increase in the number of molecules aligning to each part of the reference, like this part of chromosome 8 here, we'll detect that as a duplication. Similarly, a decrease in the number of molecules is called as a deletion. And this tool will detect large events starting at 500 kb all the way up to entire aneuploidies. Now, if you're looking at a cancer sample that is very heterogeneous, we want to use yet another pipeline, and that is our rare variant pipeline. And the way this works is by aligning single molecules directly to the reference. When we detect a structural variant, for example, in this case, these labels align and these labels align, but these labels from the reference here are missing. So this is a deletion. When we detect these structural variants, we will take these molecules, do a local assembly, again, to calculate the average spacing, and then detect these structural variants based on that. And because this is a single molecule analysis method, and because our instrument can collect so much data, we can detect structural variants down to just 1% variant allele fraction. What that means is that if 2% of the cells in your sample have a heterozygous translocation, say, we will still pick that up. Now, because we're looking at single molecules, these measurements are slightly less accurate, and so we look at a bit larger variants starting at about 5 kb here. But 1% or just even 5% allele fraction, genome-wide, unbiased, without any amplification, without any panel, that is just not possible with any other method. No one else can do this. Once the genome is analyzed, it's presented to you as a circus plot where translocations are shown in the middle, copy number tool is shown in this first circle, and then structural variants shown outside of that. You can click on any variant to take you straight to a map that shows you the alignments, and you can even see the molecules, if you like, that were detected to call that structural variant. And if you don't want to use our visualization software, you can export these structural variants as a standard VCF file and use any software you may have that can handle that. Now, what's important is that our software comes with a built-in control database of several hundred human genomes. And the reason why that matters is that it allows you to filter down these structural variants extremely easily in one click. So here's a normal sample. This could be my genome, say, and we call a total of about 5,000 or so structural variants, a little more than that, 5,600 here. And if you look at the circus plot, it's mostly the green and the orange dots. Those are insertions and deletions. We see no translocations in a human genome without any major genetic abnormalities. Now, with a single click, we can get rid of all the structural variants that we have seen before in that control database. And then this picture looks like that. So again, I'll show you without filtering. With filtering, you see that from 5,000 structural variants or so, we go down to just 50 or so that are unique to this sample, simply by choosing variants not present in a control database. And we can use the same tool to take a cancer sample and get rid of normal population control variation to zoom in those somatic variants only. And I'll show you a paper that worked on that. Now, how does Bionano compare to other methods? Well, sequencing technologies just cannot detect these structural variants very well. Short read sequencing with Illumina has very poor performance for structural variants. That's why all these other methods exist in the first place. With long read sequencing, typically you can pick up some structural variants, but the sensitivity is poorer than ours. Long and complex structural variants are missed, and read lengths are a whole lot shorter than with Bionano. With nanopore sequencing, the SV detection has such low sensitivity, and you have so many false positives, so a low PPV as well, that it's just unusable for whole genome detection. And for all these long read sequencing technologies, the output and throughput is just unusable for cancer, where you want to collect an enormous amount of coverage. So compare that with Bionano, our Sapphire instrument, where we image 300 kb molecules, where we can get 400 exome cancer samples on 3 samples per day per instrument, extremely high positive predictive value, meaning that our structural variants that we detect are actually real. And as I mentioned, you can go as low as 1% variant allele fraction with 1600x, completely in a different league than sequencing-based technologies for SVs. So this is really the technical introduction that I wanted to show you today. From now on, I'm going to show you examples of discovery research, of cytogenomic studies. But many of the examples I show you were presented at our next generation cytogenomics symposium that we organized in January of this year, 2021. All of the presentations from that symposium are available on our website, and I'll show you at the end where to find them. But keep that in mind. Every example that I'll show you is just a tiny little bit of a flavor for a talk from our symposium that's on our website. Now, let me start with discovery research. The first case I want to show you is from a patient with congenital diaphragmatic hernia. It's a very severe congenital disorder where there's a hole in the diaphragm and the developing intestines push into the developing chest cavity and cause abnormalities of the lung and sometimes the heart. It's a very severe disease. And the team at Harvard Medical School has been using Bionano Sapphire instrument to look at these children and trying to find new candidate genes that can explain this disease. Now, in this particular case, a 2 megabase pair duplication was picked up by chromosomal microarray. But when we analyzed this with Sapphire, we saw 2 separate duplications. Now, when you look at the alignments here, you can see that this section of the reference genomes aligns here to the left, but also here to the right. And this section here aligns to the right, but is also in an inverted duplication in between these 2 copies. We know exactly which genes are present and are potentially affected by this duplication. So an enormous amount more information than was available with the array, because we add order and orientation of these duplications to it. Dr. Francis Hai, who leads this study, is actually presenting a webinar March 24th of this year. You can find more information about this on our website, but please make sure to watch this because they have data now on over 100 patients and parents. So it'll be a really exciting presentation. Here's another example of how our optical genome mapping technology can find variants that are missed by other methods. In this case, because the gene is camouflaged. Camouflaged means that it's invisible by NGS, and that typically happens when there are repeats involved or when the GC content is too high or too low. In this particular case, in this important Alzheimer gene, there is a variable repeat binding domain, meaning that some of these exons are replicated, and therefore the short read data cannot be aligned correctly. Bionano doesn't have that problem because of our long molecule, so we picked up this deletion in this region, which may be associated with Alzheimer's disease. This is a study from Mark Ebert at Mayo Clinic, and there's a great webinar by him on our website as well. And another study from Mark Ebert was on ALS. In ALS, there is a repeat expansion in this gene called C9orf72, and that expansion can be tens of kbs in length. With standard molecular methods, it is absolutely impossible to size that repeat correctly because it becomes so long. But here you see multiple maps that have this insertion starting at 2 kb all the way up to 32 kb. And because we have a single molecule method, you can actually see the number of molecules that align to each of these different insertion sizes. This is a single brain sample from a patient who died from ALS. And so you see this mosaicism in the brain shown really beautifully here. Yet another example of a variant missed by other methods. Here's a study on uveal melanoma, melanoma of the eye, and a study by the Curie Institute in Paris. And Dr. Gentien there was expecting to find a variant in this gene that is typical in this form of melanoma. But with NGS, they were unable to detect any variant at all. With Bionano, they picked up a 740 base pair deletion that overlapped nicely with this part of the genome that has very high GC content. So because of the high GC content, the sequencing there failed and was not able to pick up that deletion in the promoter. Now, a team at Children's Hospital of Philadelphia has worked with partners all around the world to study the George syndrome, or 22q11.2 microdeletion syndrome. This is a genetic disease caused by the recombining of segmental duplications, these very, very large repetitive segments that can span hundreds of kilobase pairs in length. And standard methods, any even long-read sequencing methods, cannot sequence through these segmental duplications. But because of Bionano's megabase size molecules, they were able to perfectly characterize these complex repeats on single molecules and learn a lot about the variants that may or may not cause this disease in the patients in the first place. And segmental duplications being very long repeats are one part. There's also shorter repeats that matter. One example is fragile X mental retardation, which is caused by a repeat expansion in the FMR1 gene. It is standard of care that in a child with developmental delay or intellectual disability, a chromosomal microarray is run to look at structural variation, and a fragile X test is run separately as well. With optical genome mapping, we can perfectly detect the expansion here. This is a total of about 1,000 base pairs expansion in the FMR1 gene. So again, this allows you to really get the data that require multiple tests right now in one. This is a repeat expansion. There are also repeat contraction disorders. And FSHD, or facioscapular humeral muscular dystrophy, is such a disease that is caused by the contraction of a large repeat called D4Z4 near the telomere of chromosome 4. When this repeat contracts below a certain size and you have the permissive 4qa allele, you can get the disease. If you have the 4qb allele, or the repeat does not contract, then you do not have the disease. Now, genotyping this is extremely complicated, No molecular method, modern molecular method can do this. So the standard of care here is to do this with Southern blot using radioactive labeling. It's slow, it's very labor intensive. And did I mention radioactive labeling? Labs just want to stop doing that. So with Bionano, as you can see, genotyping this takes seconds of analysis. You simply look at these variants and you see what's going on. And to make this even simpler, we developed a custom piece of software that will call Bionano and Focus FSHD, which genotypes this locus for you. It will size the repeat, it will genotype the permissive or non-permissive allele, and look for a number of other markers related to this disease, and generate a report with quality metrics and all that. Based on this success, PerkinElmer Genomics and the University of Iowa, which is the largest testing site for FSHD in the United States, have launched clinical tests based on this and Focus FSHD program. And there was an incredible talk by Aaron Bossler, who heads up the University of Iowa, at our symposium on this, which I highly recommend you watch. Now, of course, 2020 and 2021 were dominated by COVID-19, and there was, it became clear very quickly that there was a big difference in how different patients responded to infection. Some people would get really very ill, and others were asymptomatic. And so Dr. Ravi Kolhe from Augusta University brought together a consortium of scientists to look at large structural variants in the genome of the patient. So we're not talking about the virus, but we're talking about the patients themselves that may explain why they did or did not get sick. He also presented this at our symposium. We actually had an entire day about this. But a paper in medRxiv gives you the highlights of some of the findings of the consortium on the first 37 patients that were all extremely ill, ICU admitted. In this study, they identified 11 structural variants, of which 7 were rare or unique to specific patients, and 4 were common that may predispose to severe illness with COVID-19 when you get infected. And these structural variants affect genes in 3 different pathways: innate immunity and inflammatory response, airway resistance to pathogens, and then viral replication and spread. And if you look at this figure, you kind of see these different parts. The green and red boxes are genes affected by structural variants detected in this study. You see that they affect interferon production. You see that they affect cell-to-cell viral transmission, and also T-cell activation and viral replication. Let me show you a few examples. Here's a case of a duplication that was detected, and you see how beautifully these maps here show this duplication affecting one of these genes involved in cell-to-cell viral transmission. Here's an inversion that affects a gene involved in the mucosal function and is important in allowing the entry of that virus into the lungs cells. And then here's a duplication again that affects the STK26 gene, which was found in a patient who was severely ill and was found to be upregulated in really all the ill patients as well. So really very interesting finding. So it's just incredible to see that even the most widely studied disease of the last year still has a lot of secrets that can be resolved with our optical genome mapping platform. Now, I'm a big fan of this study led by a team at UCSF because they studied patients, children, with a variety of rare genetic disorders. And what's interesting here was that these children were already tested with the standard of care in the clinic, which is typically chromosomal microarray and whole exome sequencing. And they were negative for both of these tests. So now in this study, they analyzed the entire genome. They used 10x Genomics linked reads, which is now discontinued, to look for single base pair changes, and then optical mapping with Sapphire to look for structural variants. And they were able to increase the diagnosis of these undiagnosed patients in 40% of the cases, and in many more, there were strong candidates. And when you distinguish between those variants detected by each method, we found that 18% of these additional diagnoses came from Bionano alone. So what's important to realize is, as I'll show you in this section on cytogenomics, is that Bionano already can detect these structural variants that are picked up by array. So this 18% are on top of what array already could detect. So Bionano could really do both of those. Just incredible result. And I'll show you two examples here that Dr. Kwok, Pui Kwok, presented in a webinar, again, on our website. And here's a case of a family where a duplication was detected in this girl, a 32 kb duplication, and this type of event was missed by array because it's too small, and NGS can just not piece these together typically. And then in another family, in a child with neuroblastoma and developmental delay, de novo translocation was detected where Bionano perfectly identified the reciprocal translocations. Now let's switch from genetic disease to cancer. We have a lot of examples of discovery research in cancer as well. And this study just came out by a team led by Jim Broach, who heads up personalized medicine at Penn State University in Hershey. And his team looked at a large number of different solid tumors with optical genome mapping. And we had a lot of data already on leukemias, hemalignancies, but not so much data on solid tumor. And this study clearly showed that high quality structural variation data was collected very easily on this wide variety of tumors, from ovary, lung, liver, kidney, colon to breast and brain and bladder. And importantly, what they found is that using this control database that I've shown you earlier, you can simply get rid of the population variants and only show the somatic variants that are specifically to that tumor very efficiently. And what's also important to see is that just in a fraction of a second, you can scan through these circus plots here and see that some of these samples, like the thyroid samples, just have very stable genomes with hardly any translocations, while, for example, here, the ovary, prostate, and brain just have an enormous amount of translocations. It's very easy to get to this type of detail in seconds with just a few clicks. These circus plots are automatically generated by our system. Optical genome mapping is a streamlined tool for precision medicine. I'll talk about this study more in a little bit, but what's important here is that you can see a three-way Philadelphia chromosome, which is really the first important personalized medicine variant that was detected, the Philadelphia chromosome, but here in a three-way chromosome, and you can just very easily in seconds see what is going on, translocation between chromosome 9, 14, and 22. You can also detect new fusions. In this particular case, BCR, which is one of the translocation partners for the Philadelphia chromosome, is fused with XOC2, which is a variant that is not typically seen in leukemias. This is a study on 48 leukemia samples that I'll tell you more about in a minute. You can detect important fusions like NTRK fusions. This is a type of structural variant that is present in many different solid tumors, but in a low percentage of the patient population. So if you can detect this, there's excellent drugs available that can help you if you have this tumor. But it's very hard to find these patients, and our method is great for that. In this study from the Cordelier Research Center in Paris, the team was looking at hepatocellular carcinomas, and these two circus plots here are from two different tumors, and you can see this very obvious difference between this fairly stable genome and this tumor sample that is a highly rearranged copy number profile, and then of course a large number of translocations. And the difference between these two tumors is that the one on the right has an insertion of the hepatitis B virus, which is shown here, just upstream of the cyclin E1 gene. And so this creates a mutational signature by creating replication stress, because the cell is forced to replicate faster than the DNA can catch up, and that causes this signature. This is a very important signature because it could mean that this tumor could be responsive to PARP inhibitors, for example. And so there's a whole new application there for these drugs if you can detect its signature. And then I want to touch on gene editing real quick. CAR T cells are an important treatment option in case of cancer. And here we looked at a gene editing study, and there was a cut made with CRISPR-Cas9 in the long arm on this chromosome, and we detected the cell fusion of these long arm head-to-head, as shown here in this cartoon. On the map, you can see that this part of the reference aligns straight to this sample map, but also in an inverted duplication right here, and that is this head-to-head fusion. We collected a lot of coverage and were able to detect this event at very low allele fractions. So this could be used to QC, genome-edited cell lines, or CAR T cells. And then I want to touch on two groups, one at UCSD here led by Paul Michel, where a team led by Vineet Bhavna developed custom software to combine optical genome mapping data with NGS data to look at very complex cancer structures, in this case, extrachromosomal circular DNA. These are the little, almost plasmid-like pieces of chromosomes inside cancer cells that can be used by these cancer cells for massive amplification of oncogenes. And in this particular case, in a glioblastoma cell and in a large cell lung cancer sample, they were able to reconstruct this circular DNA, in this case, including a MYC amplification, just really beautifully using these long maps. And then somewhat similarly, a team at Weill Cornell led by Marcin Imilinski used NGS and optical genome map data to identify novel complex structural variants in cancer genomes. And they named them after specific Greek terms, including this pattern here, named after, called Typhona or Typhoon, because it kind of looks a little bit like that. And you see here this translocation, an enormous number of translocations really weaving back and forth between these two chromosomes, involving a MYC amplification. Piecing together an alignment graph like this without long read data like ours is just virtually impossible. So this is some incredible work that they've done. And now that brings me to the second category I wanted to talk about, and that is cytogenomics. Now, this figure was put together by one of our customers at the NHS in Scotland, and it shows how the current standard for structural variation detection in the clinic, called cytogenetics or cytogenomics, really involves a whole lot of different processes and techniques and instruments and equipment to get to this structural variation data. Depending on the sample type, depending on whether cell culture or not is possible or necessary, a whole complex workflow is necessary to find all these SVs. And they compared that to the Bionano workflow, where no matter what the sample type, a similar DNA extraction is performed and a similar analysis can be done that shows you the same amount of data from all of these variants. Now, that was the theory. Let me show you how this actually worked out in reality. So what's important to realize is that when we're talking about structural variants, there's just a large number of different classes. There's aneuploidies, where chromosomes are duplicated or deleted. There's copy number variants that are really large deletions or duplications. Then there's the more standard structural variants of the different types, repeat expansions, repeat contractions. And Sapphire, Bionano Optical Genome Mapping, is able to detect all of them. The only variants that we miss right now is loss of heterozygosity, but we're working on that for a future software release. And of course, single nucleotide variants, which we just do not detect. But the mere fact that all of these different classes of structural variants are able to be detected with a single technology is just unheard of. Now, I want to show you a couple of different studies where optical genome mapping was compared against the standard of care. The first study is by a European consortium where constitutional cases were analyzed. Constitutional meaning genetic disease, and that were prenatal samples, children with genetic disease, and then also infertile adults. This is a collaboration between 3 groups in France and a Radboud Center in the Netherlands. And what they found was 100% concordance with the standard of care methods, and that is karyotyping, fish sometimes, and chromosomal microarray. And all of these different types of structural variants were all detected by Sapphire without exception. At Augusta University, Dr. Ravi Kohli is developing an LDT for prenatal testing as well, using optical genome mapping. LDT means the laboratory developed test, and that is really a clinical test available to patients. He gave a presentation at our symposium that I highly recommend you look. I can't do it justice here, but he showed very clearly how optical genome mapping detected all these structural variants, 100% concordance actually, that were identified with the standard of care, which is again this combination of different methods. But on top of that, Sapphire also added significant new clinically actionable information, which really means variants missed by these standard methods. And on top of that, Sapphire has a reduced hands-on time, faster turnaround time, and is cost-effective. There's also a commentary on prenatal testing that just came out in medRxiv, so have a look at that. Now, let's switch over to heme malignancies and cancer. The first study I want to show you is from Radboud University in the Netherlands, where 48 leukemia patients with a variety of different diseases were tested using optical genome mapping. And I'll show you a couple examples, but the title already points out that, again, there was 100% concordance between the standard of care, which is karyotyping, and a number of FISH tests and microarray combined for complex cases and simple cases. So here's an example of an IGH translocation, which is a very common one in leukemias and typically detected with a break-apart FISH probe, where you know that there's a translocation, but you don't know what the translocation partner is. And then you need to go fishing really to identify which gene that is. With Bionano, of course, you immediately find that translocation partner. They show an example of chromothripsis, which is really where a chromosome basically explodes and gets pieced together in an incorrect order. And again, Bionano was able to unambiguously detect that and analyze that structure in sometimes hundreds or thousands of pieces. I already showed you the slide of the BCR fusion with a gene that isn't typically seen or was never before seen in leukemias, and a total of 19 of these novel fusions were detected. So an enormous amount of room for discovery. Now, the biggest study on leukemia was published recently here from a multi-center collaboration between the cytogenetic leaders of big institutions, such as Columbia University, where Bryn Levy works, who ran the study, the Mayo Clinic, Penn State University, the PATH Group, the Legacy Health Group, Augusta University, Fred Hutch Cancer Center, and MD Anderson all got together to look at 100 AML samples. And those were analyzed in clinical cytolabs with karyotyping and followed up with some other methods, again, when needed. Incredibly, again, Sapphire detected 100% of all these clinically relevant variants in 100 samples. And on top of that, in 1 out of 4 cases, it assembled a more complete and accurate karyotype by finding relevant SVs that were missed or by refining the structure. And based on these results, they recommended that optical genome mapping be considered as a first-line test for the detection and identification of clinically relevant SVs. A team at MD Anderson, led by Dr. Rashmi Kanagal-Shamana, looked at optical genome mapping on myelodysplastic syndrome samples. MDS is a precursor to AML leukemia. And what they found was, again, 100% concordance, so all clinically relevant structural variants were detected. But on top of that, in 1/3 of the patients, smaller aberrations were found that were missed by karyotyping, that were too small for karyotyping. And because of the high resolution data, they were able to detect specifically which important genes were affected by these structural variants. So they concluded that optical genome mapping is a potent single platform assay for high throughput and accurate identification of clinically important chromosomal variants. Now in solid tumor, Dr. Kohli is also working on an LDT here using optical genome mapping. And in his talk at our symposium meeting, he showed a couple of really interesting results. Here you see a comparison between OncoScan, which is the microarray platform that is somewhat a standard in solid tumor, and the copy number tool with Bionano Sapphire instrument, where you perfectly see the same type of decreases and increases in copy number as you see with the OncoScan. And then on top of that, optical genome mapping identified many gene fusions and a whole lot of genes affected by structural variants in these glioblastoma samples. So Dr. Colley concluded that there was 100% concordance between genome mapping and the standard of care array, and that it has the potential to identify unique genomic abnormalities that are missed by these other methods and better characterize the SVs that are detected. And it also provides a better characterization of chromathriptic events like the ones I've shown you a minute ago. So to summarize, I want to remind you that our optical genome mapping technology can detect all types of structural variants starting at 500 base pairs with a sensitivity as high as 99% and a PPV of 97%, meaning that the structural variants that we detect are really there. We can get as much as 1600x coverage per sample, and that allows you to detect structural variants down to 1% allele fraction. Now, we wanted to make it easier than ever to give you access to Bionano data, and there's 3 ways to do that. You can simply purchase the system, and once you own the system, the cost per genome starts at about $450. Of course, this varies between different countries depending on currency, exchange rate, and also import regulations. You can also get the consumables, which is a program that works in many places around the world, where you simply purchase 120 genomes worth of consumables, and we'll make sure you have an instrument on site that you can use to run these samples without having to buy it. And we also offer a service where you can simply send us your samples and get the filtered structural variants back. And with that, I wanted to remind you again of our next generation Cytogenomics symposium that took place in January, and that all the 33 talks that were presented there were available on our website. So please have a look at the resources library section at the videos, and you will see the videos organized per day and also individual talks if you click at them, including, for example, this one here from the study, the multi-center evaluation on AML. So please check this out, because what I've shown you is really just a preview. And with that, I'd be happy to answer any of the questions that you were able to enter in the Q&A box.